Today I will be presenting a paper titled MDCS-MoAME, Multi-Directional Composite Scanning with Mixture of Attention and MAMBA Experts for Cancer. Survival Prediction. This research focuses on the direction of multi-MoAME survival prediction. Let's start with the background. In this work, we utilize two specific modalities: whole slide images WS-IS and genomic data. First, regarding WS-IS. These are high-resolution digital pathological images containing billions of pixels. A key feature is that they allow us to observe distinct characteristics at different magnifications. As shown in the diagram on the right, WS-IS have a hierarchical structure, with magnifications like 1x, 10x, and 40x. We can extract features at varying levels of granularity depending on the magnification. The figure below illustrates a concrete example of this. Next, for the genomic data. Genomic profiles provide critical expression levels regarding cancer cell states and immune factors, which significantly influence dynamic prognostic outcomes. In our input, this data is recorded as a 2D table, representing the specific expression level of each gene for every patient. Therefore, our objective is to leverage these two modalities to accurately predict patient survival outcomes. To motivate our work, let's first look at the limitations of existing methods. First, the intrinsic hierarchical structure of WS's Fig.A is often underutilized, resulting in insufficient exploitation of image information. Second, the gigapixel resolution of WS is Fig.B. C hinders existing methods from adequately capturing long-range dependencies within the WSI, as illustrated in Fig.B. Conventional methods typically employ a horizontal scanning serialization, which disrupts the original spatial structure of the WSI. To address this, we propose several alternative scanning patterns, specifically horizontal vertical, left oblique, right oblique, and loop-back scanning. These allow us to fully extract WSI features and effectively capture long-range dependencies. Third, current approaches typically categorize genes into 6 major classes, Fig.D, E, leading to functional redundancy and making it difficult to capture complex intra-genomic relationships. Therefore, in addition to the conventional forward scanning, we introduce an interval scanning strategy. This is designed to uncover potential correlations and latent associations among distant and sparse gene groups. Finally, the heterogeneity between modalities complicates the modeling of intermodal correlations. Although attention, or MAMBA-based models can achieve sufficient intermodal interaction from their respective perspectives, inflexible and simplistic feature fusion designs may introduce redundancy, thereby limiting the ability to improve feature representations. So, to summarize the key challenges, the gigapixel resolution of pathology slides and the high dimensionality of genomic profiles impede effective feature representation. Furthermore, there is significant heterogeneity between the pathological image and genomic data modalities. Figure A illustrates our overall framework. In the feature extraction stage, we input 2 levels of WSI features, specifically the region level and patch level features. Along with the genomic profiles, we utilize ResNet 50 and an MLP for preliminary feature extraction. Following this, we employ multi-directional scanning and feature enhancement to achieve exhaustive feature extraction. The specific details of this process are shown in Figure E, which I will elaborate on later. The resulting high-dimensional features are then processed via expert-driven intermodal interaction for cross-modal fusion, as shown in Figure F. We introduce an expert network based on attention and MoAME for modality fusion, effectively minimizing intermodal heterogeneity. Finally, to mitigate redundancy between modalities, we introduce feature alignment and prediction. Here, features are aligned via L1 distance minimization to prevent bias in the fusion results. In summary, by combining a multi-directional scanning strategy, an expert network, and feature alignment, our method effectively extracts modality specific features and mitigates modality heterogeneity.